Hello. In this short presentation, I'm gonna also to start the vascular system, which is the second part of the cardiovascular system. So we'll talk about arteries, arterioles, veins and venules, and capillaries. So let us get started. The blood vessels, as you know and as you said, they are the organs of the cardiovascular system, and the vessels form a closed circuit. As you know that the blood ejected from the right side of the heart into the lungs through the arteries and come back to the atrium through pulmonary veins. And then from the left side of the heart ejected to the systemic circulation through arteries, and then come back again to the right side of the heart or right atrium through veins. So it is a closed circuit that transport blood from the heart to the body, cells or lungs, and back to the heart. In this presentation, in the body in general, we have like 4 levels of blood vessels. We have arteries that carries blood away from the ventricles of the heart. Then we have arterioles that receive blood from the arteries. So they are like divisions of arteries at the level of organs, which is a smaller level. And then at the level of tissues inside the organs, we have the capillaries, and these capillaries are the sites of exchange, and they connect the arterioles to the venules on the other side. And the venules receives blood from the capillaries and conduct it into veins, and the veins receives the blood from the venules and carry this blood back to the atria of the heart. Um, we have some clinical applications. We have the term of angiogenesis. Angio means blood vessels, and genesis means formation, new formation. So angiogenesis means new blood vessels formation, new formation of blood vessels. Okay, and this is controlled and regulated by a substance which is called vascular endothelium or endothelial growth factor. And this substance is secreted whenever there is obstruction, blockage into one of the vessels. Okay, so it occurs in response to blockage of coronary artery, and the heart aiming to create like a bypass for this blocked vessel, just to allow the flow of the blood to pursue and to continue to nourish the heart. And it can also occur uh, as a consequence and in association with new growth of tissues or tumors. So in case of tumors, the tumors secrete the same substance, which is vascular endothelial growth factor, uh, and the tumors also aiming to sustain sufficient and continuous blood supply for the newly growing tissue cells. So in both cases, we will have what is called angiogenesis. Okay, let us compare and study the anatomy or histology of the arteries and veins. So as you see here, this is a picture showing a vein in a slide or under the microscope, and this is a picture of an artery. So let us compare between both of them. Both of them, they have 3 layers. They are made of 3 layers. The innermost layer, we call it tunica uh, interna, here and here, and the middle we have tunica media, and the outside we have tunica externa. Okay, so let us study each one and try to compare and contrast between the arteries and veins, the histology of arteries and veins. So in the tunica interna, it is almost similar in both arteries and veins. Both of them made or lined by simple squamous epithelium. But the difference, as we can see here, in the veins we have what is called valves, okay? And these valves are found mainly in the lower limb veins because the blood flow goes from the lower limbs towards the heart against the gravity. So, and there is no pump that eject this blood towards the heart. So, usually the blood moves from one area to the other area which is above, and immediately after crossing, these valves will close to prevent the backflow of blood to the lower limbs or to the lower sides. Okay, so the tunica interna are similar in terms of lining epithelium, simple squamous epithelium, but in the veins we have valves. When we go to tunica media, which is mainly made of smooth muscle, we find that it is very big and large, thick in the wall of arteries, while small and thin in the walls of veins. Okay, that is why uh, whenever we go and see the arteries and veins in the cadavers in the dissection room, we'll find the arteries thick and always the lumen of the arteries are patent, not like in case of veins, in most of the cases it is collapse. Okay, so the tunica media is thick in arteries and it is made of smooth muscles. Okay, and the third and outer layer, which is tunica externa, it is made of just connective tissue and is more or less the same between arteries and veins. Okay, so the main two differences is the valves in the tunica interna in the veins and the thick wall of the smooth muscle in the tunica media of the arteries. Okay, so this is a description of arteries. We talk about the three layers, tunica interna, media and externa. And generally speaking, the arteries, they transport the blood under high blood pressure because they receive the blood directly from the ventricles, right and left ventricles of the heart. And they give rise to smaller arterioles. And the arterioles, they have also the same three layers, the tunics. Both arteries and arterioles can undergo vasodilatation and vasoconstriction. Okay, so this is a picture showing a small arteriole. So here, this is the tunica interna made of squamous epithelium. This is the tunica media made of smooth muscle and adventitia surrounding the whole vessel from outside. And as you see here, as it goes down or up away from the heart towards the organs, the arteries and arterioles are going to divide and divide, giving rise to smaller capillaries at the level of tissues inside the organs. The capillaries, these are the smallest diameter of blood vessels in general, and they connect, as we said, the arterioles to the venules, and they are mainly or only made of just simple squamous epithelium. There is no only the tunica interna. There is no tunica media or tunica externa. Okay, so simple squamous epithelium, and why? Because they are permeable or semi-permeable to exchange of substances between the blood and the tissues. At the level of capillaries, due to hydrostatic pressure, water and oxygen are secreted to the outside of the tissues, and carbon dioxide and some water, they go back to the end of the capillaries towards the venous end. So, and also the hormones and other substances are secreted at this level. Okay, so why? The capillaries are just made of simple squamous epithelium because of this function, which is exchange of substances between the blood and tissues. Okay, so capillary blood flow regulated mainly by a sphincter. Okay, this sphincter is made of a smooth muscle at the beginning of each arteriole, capillary, at the end of arteriole and beginning of capillary. So let us see this picture, which show in this side here, this is arteriole, okay, or artery, and this is arteriole, and here these are the capillaries. These are venules, and this is a vein. Okay, so here this is a site of capillaries, and as you see, the capillary has like two ends. One end towards the arteriole, which is still looks like arteriole, and this end, which is similar to venule, and we'll see it in minutes. We'll see the differences inside uh, uh, the capillary sections. Okay, yeah, this is a smooth muscle sphincter, which is found at the end of the arteriole and the beginning of the venule, which controls and regulates the capillary exchange of substances with the tissues and the pressure inside the capillaries. Okay, let us talk about the capillary permeability. This is mainly due to openings uh, in the capillaries, okay? And the size of these openings varies according to the tissues. Some tissues that are small, others medium, and third tissue like the liver and spleen, they have very big openings. So mostly there is a huge exchange between the capillaries and the liver and spleen, for example. Also, this capillary permeability depends on the function and status of the organ. So, for example, the GI after a meal will have more blood goes to the GI system. Okay, why? Because we need more blood for the process of uh, digestion and so on and so forth. Okay, so we have like three types of capillaries. We have continuous capillaries, and these are characterized by having small openings, and we can find it in the muscles or skin or nervous tissue. We have the fenestrated capillaries, which have larger, a little bit larger openings than the continuous capillaries, and these can be found in the endocrine system and the kidneys. Okay, and finally, the third type of capillaries, which is sinusoidal. Sinusoidal, these are characterized, as I said, by large openings. Okay, and are mainly found in the liver, spleen, and bone marrow. You see in this picture, and this is continuous capillaries because now the openings are small openings between the squamous cells that form the wall of capillaries. But here, this is a fenestrated type where we can see a little bit bigger fenestrations, and this is a third type, which is uh, sinusoidal, where we can see large cavities between the capillaries and the tissues, especially the bone marrow, liver, and spleen. As I said, these capillaries, wherever we have organs that they have a high metabolic rate, we need more blood, we need more exchange between the blood capillaries and the tissues. Okay, so like in case of uh, during exercise or after meal. During exercise, we need more blood to go to the muscles, and after meals, we need also more blood to go to the digestive system. And as I said earlier, all these processes and the blood flow in the capillaries and the exchange are guarded and regulated by a smooth muscle, which is found at the beginning of the capillaries. So as you see in this diagram, also this is an arteriole and this is a venule, and here this is capillaries. lots of capillaries. Their number depend on also the function of the organ. So if this organ has high metabolic rate, then we expect to have more capillaries, okay? Exchange between capillaries and the tissues, this occur through different methods, mainly four methods, okay? We have diffusion, and we talk about diffusion in A and B1. If you remember, diffusion means the substances they move between the capillaries and the tissues, and we have two ways. If this substance is lipid soluble, then it can penetrate through the capillaries easily because the walls of the cells are mainly made of lipids and cholesterol, and the water substances they diffuse through channels, okay? So this is diffusion, and this is the most important method of transfer between the capillaries and tissues. The other method is filtration, and filtration is mainly due to hydrostatic pressure. Hydrostatic pressure, this is due to blood pressure or ventricular pressure, okay? So pressure inside the capillaries, which lead to filtration of substances from the capillaries to the tissues. The third type of exchange between capillaries and tissues is osmosis. And in this case, there is high osmotic pressure inside the capillaries, especially towards the venular side. Okay, and this pressure, hydrostatic pressure, oncotic pressure or osmotic pressure is mainly due to presence of plasma proteins because after initially at the beginning of the capillaries more water is excreted, okay? So um, the ratio of 55 plasma and 45 blood cells will a little bit affected because now more fluid is excreted, so the 55% decreased. That means the concentration inside the capillaries will increase. And this will lead to high colloid osmotic pressure, and this high colloid osmotic pressure will bring back the water at the end of the capillaries towards the venular sites to get inside the capillaries, okay? So this is the third type of exchange uh, in the capillaries and between the capillaries and tissues, okay? And as I said, at the arterial end, the capillary of the capillary water and other substances leave the capillary by filtration or by hydrostatic pressure because it is higher than the colloid oncotic pressure. At the venular end of the capillaries, the opposite occur, which is more water reabsorbed into the capillaries because the colloid osmotic pressure is higher than the hydrostatic pressure. Okay, so more fluid leave the capillaries at arterial end and return at the venous end. And as you know that the remaining amount of water and other fluids that could not get into the venous end of the capillaries will follow and access the lymphatic vessels back into the venous system. This is very clear in this diagram. This is a capillary, okay? This capillary now red here because it is more closer to the arterioles, and here become like blue because it is more closer to venules. As you see here, in the capillary there is a pressure which is called hydrostatic pressure or a blood pressure due to the pressure coming from the ventricles. So this will lead to excretion of water and oxygen, okay? Those oxygens will go to the tissues and other substances, okay? Other pressure, which is oncotic, which is pulling back the fluid towards the capillaries, but here it is less than the hydrostatic pressures. That is why most of the fluid and oxygen goes to the outside rather than getting back into the inside. But on the other end of the capillary, the opposite is correct. Here, the oncotic pressure become higher. Why? Because oncotic pressure is mainly due to proteins, cells, blood cells, which are found here. So when we lose some water, the concentration will become increase here, will become high. So this oncotic pressure will reabsorb back the water and the carbon dioxide to the capillaries. But still, there are some water that cannot get in here, so they will find their way into the lymphatic. So the lymphatic usually started at the level of capillaries, and the lymphatic drain back into the venous system. And the fourth and last way of exchange between capillaries and tissues is transcytosis. And transcytosis is occur or due to or in case of large substances like antibodies or lipoproteins. Okay. Okay, we talk about arteries, arterioles, and capillaries. Now let us proceed and continue our description to the blood vessels, the organs of the cardiovascular system, and talk about the venules. Venules, we said that they are microscopic vessels that transport blood from the capillaries into the veins, and they are thinner than the arterioles. The veins are also thinner, but still they have the three types of tonics, and we said that they have valves in the inner wall. The tunica media is smaller, not not similar to the arteries. Okay, and they ensure one way of blood flow. As you see here, these are the valves. They ensure one way of blood flow. After the blood squeeze up, the valves close to prevent backflow of blood to the lower part. And in general, the venules act like a reservoir, and that is why about 2%, two-thirds of the blood is found in the venules. Okay, 10 to 12 is found in the lungs, 8 to 11 found in the heart, and 10 to 12 found in the systemic circulation, the arteries. Okay, but most of the blood is found in the large veins and the venules, so they act like a reservoir for the blood. Regarding the blood, we have many diseases associated with blood vessels like atherosclerosis, and atherosclerosis is characterized by the position of cholesterol in the arteries and arterioles. Okay, and for sure this will affect the lumen of the blood vessels, will lead to blockage of the blood vessels, and consequently ischemia in the tissues. Aneurysm, this is dilatation of the arteries, and it occurs mainly due to weaknesses in the arteries and aggravated by increased blood pressure. And this unfortunately may rupture, leading to internal bleeding. We have also a disease called phlebitis. And phlebitis means inflammation of the veins, and that can occur post injections or heavy cannulas. Okay, it is very common type of disease, but it is mild. We have varicosis, and varicosis mainly is defined as abnormal and irregular dilatation of the superficial veins, mostly in the lower limbs, in the legs. Very obvious. It occurs among those who stand for long times, and it is mainly the problem is incompetent valves, incompetent lower limb superficial venous valves.